Yeah, so I think that automation tackles already a lot of challenges. So by automation, you can reduce the staff numbers, you can reduce by this the costs. It also reduces a lot of human and other errors. So it's also more safe, and it leads to more consistent results. And challenges that are connected with implementing automation are usually the translation. This is a big issue from a research setting to a clinical setting. And this can be really challenging if the process was not designed initially for large-scale manufacturing on an automated device. So we would always recommend to put some sorts, and we do it also in-house. If you envision to bring your CAR T cell to a clinical process, that you think right from the start, keep it in mind and choose your reagents maybe you choose reagents that are also available in GMP quality for later on, and also put some thought in IPC QC right from the beginning when it comes to panel design or to assays.